would really love to facilitate a productive discussion. I'm going to hand it over to Dr. George.
Thank you, Michelle. Yeah, so Michelle asked me to moderate this research roundtable. In order to do that, I think it's helpful if everyone has a camera presence on the call, so we're having more of a conversation. I took some notes during the morning, and I put together one slide just as sort of to get us thinking about what we want to discuss. But I think give me a second here. I need to shift some things around on my computer screen. But just to give us a little sort of guidance as to what we might want to talk about. So this is just a slide, as you'll see in a moment, that just lists a few things that were brought up today in various contexts that I list here as topics. And they're not in any particular order. They're not ordered by any particular priority or preference. It's just, these are things that need to be talked about at some point. And then the question is how to prioritize things. I actually thought Terry Jo's slide, where she showed above the line were things that families need to be involved in, and below the line are things that primarily are the job of researchers. I thought that was very, very astute, and I would love to have that slide at some point. So anyway, here's just some things to think about. Some of them we just heard about from Terry Jo, which is clinical trial readiness and natural history studies. Certainly understanding the disease, which I label here as investigating the pathophysiology or the mechanism of the disease, is also important. How are we going to do that? Well, we need models. We need disease models, cell models, animal models, that sort of thing. An important question that I think is always raised with these types of neurodevelopmental disorders is the phenotype, the condition, reversible? And at what point or what age is it still reversible? And there's been some clever ways to assess that using mouse models that I'm aware of in some neurodevelopmental disorders. So that may be a topic to discuss. And then sort of pivoting to therapeutics, the idea of drug repurposing was discussed, led by Ethan, versus sort of a de novo drug discovery effort. And when drugs are to be tested for whether they are drug repurposing opportunities or new drug entities, where do you test them? Where do you test them first and second and third? Cell type, cell model, animal model, what does that look like? And then the whole emerging area of genetic types of therapies, ASOs, base editing, gene therapies. That's another topic I think that's very ripe for a little bit of discussion. And then one I think that kind of folds back to the very first topic, which is clinical trial readiness, but more importantly, clinical trial design. I think that what we've heard is that natural history data can supplant a placebo control arm in a placebo controlled trial, but this is a really rare disease. It's not as common as something like Dravet syndrome, and where placebo controlled trials were very successful. This is really rare. So I think some clever approaches to trial design need to be worked on. And then lastly, and certainly not least in its importance, what do you measure? What's the outcome? And how does that vary by individual? So anyway, I didn't list anything under priorities because I think that's partly the purpose of the conversation here. So the floor is open. I think there's no organization here other than people talking, and maybe we could just go around and hear from individuals as to what their take-home message was from today, and whether they think anything was not covered, and whether there's something really important that should be covered. So I'm looking at the chat box here. I don't think there's nothing that I need to see. So Anyway, anybody, or I'll call on somebody. That's always a very uncomfortable thing to do. So I will actually direct the first question, since we just heard from Terry Jo. And again, I think you made, a very passionate statement, which is doing these natural history studies is a pain, very challenging, very difficult. Is there any strategy to make it less painful? Any, and I know you know Ann Berg well. I've worked with Ann for a few years, and she's done a lot of work with the Clearings database to try to make these online surveys less painful. What's on the horizon to try to make this, if KCNH1 starts a natural history study this year, what's the best formula to make that as least painful as possible?
Oh, that's a great question. There are a couple different tactics that we've been trying at Combined Brain. One is to make sure that the families get a report, or as quickly as possible get results from whatever tests are done on those on their kids, and that way they can hopefully have those and use them as they see fit. So if they have developmental testing, give them the report right away so that they can actually give that to their school or some other researcher. Make it possible for them to share the research elsewhere so they don't have to do the same exact thing over and over again. So that's one thing. Another thing that we've been trying really hard at Combined Brain is to find platforms and ways for daily information on it. So rather than just collecting information at set intervals, they can actually add information that you don't even know you need to collect. So for example, if a kid falls, they can enter into a program fell today. Or if a kid has bad behavior, they could enter bit someone today, or whatever it is. And then you can go back and look at that in relation to the seizures or in relation to medication changes. And then over time, you as a research group can realize that, oh, falls are really important because we have 5 families collecting it, or constipation is a big one. Nobody ever collects that information. And it turns out that's one of the big things that impacts people's lives. So if people can use the information on a daily basis, it keeps them engaged. And you don't have to just bug them once a year to fill out the form. Instead, you can help them use it for their own lives.
Now, am I on here? Yeah. Is there any kind of smartphone app that's been developed to enable that kind of daily data collection?
There are. So there are different ways to do this. So Simons collects the standardized surveys, which is fantastic. And then if you do the visits to the actual clinic to collect in-person assays, that's really important. But we work with another platform called the Matrix. The Matrix can collect that kind of daily information, um, and collect all the other stuff as well. There are also individual apps that you can use that have been known to be good enough to be put in CDISC format so they can be used for FDA trials. So those would be things like Seizure Tracker, that's from the Epilepsy Foundation. There are diet apps. There are a lot of different apps that can be used depending on what's important to your families and your trials.
And is there a place where this information about these tools has been aggregated to make it easier to learn about them?
Well, that's sort of what Combined Brain specializes in, and we're so excited that this group is now a part of Combined Brain so that they're new. So truthfully, I don't know enough about the disorder. We haven't done a draft conceptual model, which is something we usually do for each of our disorders. We're hoping to have an undergrad on that this semester. to do our first draft so that we can start to understand the disorder a little better. And then we make recommendations after that about what we think would be really smart to collect based on what you all are already collecting, and then what hasn't been collected so far and what the families feel is really important.
So you're looking for an undergrad to do something.
Well, I teach translational neuroscience at Vanderbilt. And so what I do is I just assign an undergrad to each of the new disorders. And so we're going to have a speaker from your group coming up, I think, next week or the week after. I can't remember. And that launches the student into it. And then we take their little draft at the end of the semester and we assign it to one of our postdoctoral researchers, and they beef it up so that we know a little bit more about the disorder.
So I'll just mention that although I'm not in Nashville anymore, I mean, I was there for many years. There's an undergrad that had worked with us last summer who began to do some electrophysiology work on KCNH1, who just contacted me and said she can't come to the lab during the winter and spring quarters this year, but would like to do a remote research project. Is that somebody that could a remote project be done in tandem with a Vanderbilt undergrad?
Oh my gosh, fantastic. She could join my bevy of undergrads.
Yeah,
We'd love to take her on.
yeah. I'll contact you and get her online. She's
This is why
very bright, hardworking student. So
fantastic. This is why you all do these conferences. So we'll do these kind of connections. So thank you
good.
so much.
That's good. There are some things in the chat. I'm sorry, I have to look to my other screen, so it looks like I'm not looking at the camera. Let me see. Yeah, there was a statement here, I think very interestingly from Hans Schumann about do we need to get information about, well, I'll just summarize, drug metabolism profiles to inform about drug repurposing work. I think that's a really interesting question. And I'm curious if Hans is on the call. Can he elaborate on why he brought that up? I don't know who is on the call. There we go. Hans, if you unmute, you can just tell us where that came from.
Okay, now can you hear me?
Yes.
Yeah, so this comes from the work that I had done for many years in the modeling field, in silico modeling, on drug metabolism, and the experience that Tristan had with imipramine. It's a very difficult drug to dose, and what I'm trying to understand is are there things specific to the KCNH1 family that would make these kids fast metabolizers or slow metabolizers, things like that, which would help inform some of the drug repurposing and trying to find the right dose that's actually going to work. We know that kids in general, their liver is not the same as an adult liver. And given the developmental issues that some of these kids have, I just want to know, is that something that would be useful to collect to inform drug repurposing and natural history and say, hey, you know what? All of these kids are fast metabolizers. We don't know why, but they are. That's good information. And so I'm just trying to understand that. And the reason that I brought it up is because it seems like drug repurposing is the quickest way forward. All of these other paths have a long distance to go. And so any info on that side might be useful. And when I was listening to the conversations about collecting natural history and whatnot, that was one of the ones. I don't know if it really fits as natural history, but it was something that I was interested in, not only for one drug, but for any drug. And I just thought it'd be good information to find out if that's collected, if it's available, et cetera. So that's where I was coming from.
Yeah, it's a really astute point. And, again, I don't have any answers. I think that the approach to doing that could be challenging. It's kind of outside of routine clinical care. However, the resources for doing genetic testing of drug metabolizing pathways is becoming quite available. And in many cases, and I'm not sure this is the case for many families, but some families got a KCNH1 diagnosis because they had an exome sequencing test or a genome sequencing test. So the information about drug drug metabolizing enzymes and drug transporters and all their genetic variation is already collected. Someone has to dig it out, and I'm not sure how one would dig it out. Empirically defining drug metabolism as a phenotype would require some kind of standardized drug exposure. And I don't know that, again, I think that falls a little outside of routine clinical care. That said, again, there may be empiric observations about that, the dose given to a child was insufficient to get a therapeutic blood level, and that implies a rapid metabolism, where, independent of how it got there. So, I think that's an interesting point.
And one more comment on that is that many of these patients are already taking other therapeutics, and there is drug-drug interaction and inhibition and induction of the various P450 enzymes, et cetera. So it really, unfortunately, it gets more and more complex, but I think it's going to have a really big impact on the repurposing side of the coin.
Yeah, it does really call for it does really highlight the importance of including a clinical pharmacologist in the discussions about drug repurposing, and maybe even have that as part of the clinical trial design. Extant medications need to be looked at for whether they are drug metabolizing enzyme inducers or inhibitors, because that may in fact affect drug dosing considerably. Unfortunately, most of those things are not
side effects. Can I just share, since Tristan experienced probably most of this, I would guess that, that the gene pharmacogenetic testing is not probably a routine part, but we did pursue that for Tristan before starting on the imipramine, because we wanted to know what would be the best dose for a little guy like him, especially. And so I found that very helpful and reassuring to find out, oh, okay, so he is an intermediate slow metabolizer. So we should reduce the dose by 25%, which was the recommendation for that category. And then it was also, I'm not sure if anyone else in going in looked closely enough at his existing medications to recognize that his Prevacid that he was on also inhibits the hERG channel. And so when he did experience the prolonged QT, as after introducing, we got a baseline EKG, and then he did experience the prolonged QT after introducing the imipramine. Thankfully we did recognize that there was that potential interaction effect. And we determined at that point to withdraw the Prevacid, since it didn't seem to be helping anyways. And that did return the QT back to a more comfortable level.
That's really interesting. Yeah, so perhaps pre-trial pharmacogenomic testing and also review of medication portfolios with a clinical pharmacologist should be kept in mind. So Ethan is still on the call. I mean, Ethan, this is probably not something that's commonly done, I would think, in drug repurposing circles, is to do pharmacogenomic profiling. Have you encountered that?
It's a great question. I'm glad Hans brought it up. And I think this is channelopathies, I think, are going to be a place where this maybe becomes something of a standard, given your baseline of polypharmacology and the fact that we know that these class of drugs, like these tricyclics, have this intrinsic, polypharmacology where they're probably going to engage with these channels in some way or another. So I think it's something to, maybe that's a discrete research project of digging into the WES files and just pulling out all the CYP SNPs and then running some profile to get an assessment. I mean, 23andMe will tell you some amount of this information from those old arrays. So we definitely should be able to figure out a way to standardize this.
Yeah. Michelle, you said Tristan had some kind of genetic testing done for drug metabolism. And was that something that insurance paid for? I'm just curious.
Yeah, I mean, I had to jump through a couple hoops to get it covered. But, the pediatrician wrote a prior auth, and it wasn't too much of a problem.
Okay.
But yes, I agree theoretically, we should be able to extract that from the West, but I'm not really sure how you would go about doing that.
Yeah,
Seemed
it's not trivial. I mean, again, and companies are in the business of selling the data, and if they haven't mined it already, someone will have to mine it, and they may charge for that. So anyway, still seeing chats. Peter, you have your hand up. I'm sorry I didn't see it, but please go ahead, unmute yourself and ask your.
Yeah. So no, I just wanted to add to the conversation that just beyond also metabolizing speeds, there are just differences in the physiology of drug uptake between adults and children. And most of the patients here are children. And I know this because I'm from Holger Lerches group in Tübingen, and we recently ran this study for KCNH1 patients. And what we learned was that we had to increase the dose tenfold in comparison to the adult population in order to reach similar levels in the serum. So I think also just, adding to general drug repurposing trials, this metabolizing thing is nice, but I think you still have to measure the concentration of the drug actually in the serum itself, because there's more going on in children physiology than just like zip enzymes.
Do you have any ideas about what is causing? I'm just curious because that exact thing happened to Tristan. We introduced a certain dose, we tested his serum levels, and they were undetectable, even though theoretically they should have been reasonably high at that point. Increase the dose and still practically undetectable.
Well, we don't know. It's just that children are not just small humans, and you can't just take the dose that was calculated for an adult and that was proven to reach a certain serum concentration in an adult, and then say, okay, we adjust this now by weight, and then it will be the right dose to reach a given serum concentration. And I'm not sure whether actually there is any kind of help in these kinds of questions other than testing it.
So the important thing is maybe empirically you must really determine blood levels and do pharmacokinetics in each clinical trial sample or case. Good point. Does anyone want to address the issue of phenotype reversibility? And again, the examples that I'm familiar with all involve mouse model studies. And Jennifer is probably familiar with the SCN2A story. There's also a SYNGAP story.
Yeah, and I think obviously step one is we would need to either make a conditional model, or the other thing is to reintroduce if we can reintroduce or overexpress wild type in knock-in. But certainly I think the other part of it to consider too is there may be some phenotypes that are reversible, and there may be some phenotypes that aren't necessarily reversible, but at least you plateau, and there may be others that it's the timing is off. So I think there might not be a single intervention window. It may be a variable intervention window depending on what you're talking about.
Yeah, that's a good perspective. It's not everything is going to be the same. So yeah, I think the reversibility experiments that I was most impressed with all involve these very complex mouse models where you have a mouse that has the gene knocked in at some point, the mutation knocked in, and then it can be reversed out later. That sounds challenging.
Yeah, we're starting to work on some of those for KCNB1, which is another voltage-gated potassium channel gene that's similar to KCNH1. So yeah, but you're right, it's complicated, and there are certain mutations that are easier to do it for than others. So there's a little bit of selection that has to go into it.
Other types of animal models, again, I don't know that there's any need to think about other models other than a mouse, but there's been a trend to do things in, say, zebrafish, which may enable some in vivo drug screening, which is difficult to do in a mouse. And then beyond mouse, there is, I think there's been an effort in SynGap to make a SynGap rat, because the rats maybe have a little more robust behavioral phenotyping pathway. So any thoughts about that? Zebrafish or rat models?
Yeah, and there's also been a move to make some marmoset models for some of the neurodevelopmental disorders that lean more towards autism. So I mean, really, at the end of the day, what model you make depends on the question. And you're right, drug repurposing screen can be done with zebrafish pretty easily, as long as there's a nice correlation between one of the zebrafish genes and the human gene, which I'm not sure is as clear cut for KCNH1 as it has been for some other genes.
So I just want to talk about the reversibility issue a little bit. In the Angelman world, what we found is that certain phenotypes could be reversed completely if the gene was replaced very early on. But if the gene was replaced in the adolescent, you could not reverse motor skills or seizures, and nor some of the social issues. But even in the adult mouse model, you could still reverse the learning and memory markers. And for me, that's the holy grail. So I think we have to be really careful when we look at reversal in models where we can't measure all these subtle distinctions. And we also have to go back, like a broken record, to what the families care about.
Any other comments about phenotype reversibility? Yes, please.
Can you hear me?
Yes.
Yeah. Hey, it's Matt Weston. I know it says Amy Shore, but I got an email from Michelle the other day. Said come to the conference. So I hopped on, saw some of the stuff. Nice data. I mean, I was just going to mention, in KCNT1, which I'm most familiar with, the Petro paper recently with the ASOs showed. I was shocked how good of reversibility of phenotypes they got with the ASOs, even treating mice that were day 40 at P40 or something like that. So
yeah, so definitely can happen. And
yeah, I mean, that's closely related in some way if you consider potassium channel mostly gain of function variants that you can do. So yeah.
Can you remind me, that's a general knockdown ASO, is that right?
You mean it's not targeted to the variant? Yeah, it's just targeted to the gene.
Yeah.
Right.
Correct.
Wonderful.
Yeah, Matt, while you're on the call, I think the other question that probably needs to be answered at some point is which neuron subtype is driving the phenotype with KCNH1. And
yeah, I'd love to get involved in that. Yeah, I mean, I was shocked. I didn't know much about KCNH1, and then I got the email and I trying to look at what's known about it, and I was shocked at how little, or at least I could find without a deep dive, is known about its role in neuronal physiology. So it seems like there's a lot to learn on that level.
Right. Yeah, and again, I think getting the right tools. Again, Jennifer Kearney is starting the work to make a mouse knock-in mouse one of the more recurrent mutations. So hopefully a year from now, there'll be ample tools and opportunities to work on these things. Yeah, anytime there's a gain of function of potassium channel that potentially causes some kinds of hyperexcitable phenotypes in the nervous system, I can't help but think that it's driving or diminishing excitability of interneurons. I just can't not think that way, but it could be more complex than that.
Yeah, I mean,
But
that's what we think's going on with KCNT1. We finally have a model that we think we can use to answer those questions. And but other people think differently. And I think with KCNQ2, 3, Tasso with his model thinks maybe it's something going on in excitatory neurons. I think you saw his talk at AES. So yeah, it'll be really exciting to figure out. Yeah, it'd be great to collaborate in the mouse model with you all.
Good.
I mean, if you just have a look at the data that the Allen Brain Institute provided for single cell transcriptomics, I mean, you can see that the expression, I mean, although there is some expression in the interneurons, it's mostly in the excitatory neurons of the cortex where KCNH1 is really highly expressed. But there are a few interneuron subtypes from their map.
Yeah,
But we also,
where I think I don't know, I go back to KCNT1 because that's the only one I know, but I think like where in the cell it is can be just as important as obviously how much is expressed. And
yeah, and there can be compensatory changes in the inner neurons because the excitatory neurons aren't behaving as expected. So I think that's where the potassium channel field, there's a lot of dynamic regulation of these channels. So there may be a lot more happening than we know, and it may not all be related exactly to the KCNH1 biophysical change.
Yeah. So I mean, we have been working with single cell transcriptomic data from transgenic mice with different kinds of mutations, and it really can change in every cell type. There can be changes. So I mean, if you at some point plan also to do single cell transcriptomics, then I would also be very happy to be involved, or our lab would be very happy to be involved in that.
So let's see here. I think we just have a few minutes left. I'm just wondering if there's anything on this list that is important to discuss as a group. I think the emerging genetic and molecular therapies is certainly one. I think as Michelle mentioned in her introductory comments, she's worked with IX cells to do at least an initial screen for ASOs, non-specific, non-allele specific ASOs. Those are going to get tested in iPSL neurons in our lab at some point. We got to get the neurons made. But again, I think that the ASO pathway has been made to seem a lot faster and quicker than it is. It has got a lot of bumps in the road, and there was, as probably some on the call know, there was a well-publicized tragic fatal event in an ASO trial at Boston Children's Hospital recently, involving two kids with, or one kid in particular with KCNT1 who died of hydrocephalus, and another kid in the same trial whose hydrocephalus was detected pre-symptomatically, and they corrected it and stopped the ASO. Again, nobody knows why that happened. So there's still a lot of unknowns about these what seem to be magic bullets. But so anyway, just not trying to say not to do it. It's the road is probably a lot longer than we would like to think. What is going on with NLORM, by the way, Michelle? Is that an active project with them?
Yes, although there's been no movement for, we're just still waiting on the long read seq results, and they were supposed to be within 90 days. I think it's been like 5 months now, which seems very long to get those results back. So yeah, just waiting on that.
Is that for one particular patient or a bunch?
Yeah, no, just Tristan. They only, as far as I know, at least technically, they only accept the, single cases.
Yeah, their rubric is ultra rare diseases.
Nano rare.
Nano rare, thank you. It's nano rare. Which could be, a non-recurrent mutation in a gene that's, for which there's 500 cases.
Yeah.
But that's a unique mutation, so they will take that.
Yeah.
I think the other thing that's just worth talking about is, and I think it was Wendy Chung's comment in the very beginning that said that it's very important to rally the community and to get them engaged and to, as she put it, not splinter. And I think that was a good way of expressing the phenomenon that I think many of us have seen in rare disease communities where foundations splinter, or there's a separate foundation that gets started. So I don't know where the landscape is right now with KCNH1. I think, again, Michelle, I think you're kind of leading the charge. I don't know how engaged everyone is. Do you have a sense for that? And I certainly was hoping that maybe by doing this webinar today, there would be greater incentive and interest in being engaged community-wide. So what other things could be done to make sure that the community is fully engaged prior to setting up these very important next steps, including a natural history study?
Well, I think this was just a huge step, so I really appreciate you encouraging us to pursue it and make it happen. I appreciate Dr. Michelle's passionate speech. I hope that was effective. So yeah, I think, I think a good, I'll have to look at the registration list, but I think a pretty good percentage of the identified, or that we have access to, contactable patient population turned up today. So I'm hoping this will be a launching point for, increased engagement and involvement going forward, particularly in the natural history study. I know, as Dr. Michelle noted, they can be a pain, so I understand if people get tired of participating, but it's so important.
Yeah.
I do think that whole thing of splintering is so common, and I don't really know how to prevent it. I think it might be just almost inevitable. So what I always tell people is just keep facing forward, keep doing the right thing, keep your arms open, try to meet with everyone, don't shut anyone out. And so hopefully, even if there is a splintering, you'll still be able to work together.
Absolutely.
At Combined Brain, when we let in new disorders, we now have a policy that we let in all the organizations at the same time, and they have to agree on their votes, and they have to agree on their appointments to the committees. So it forces them to work together. So far, we don't have to do that with you all, but we've had to do it with some of the others, and it's our marriage counseling role.
Exactly.
Wonderful.
Exactly. There really isn't another group, right? I mean, there's the Facebook. We heard about the Facebook group, certainly.
Yep. Yeah, the Facebook group has been there and active for years. People are wonderful and supportive on it. But a lot of these families were like the pioneer KCNH1 families that spent the majority of their kids' lives not having a diagnosis. So it's probably a pretty extreme transition to go from never knowing what's going on to like, wow, we could actually do something about this.
Yeah, no, I think it may be that there maybe is, and I've seen this with other organizations. There is a bit of a disease fatigue, I think, that sets in, and
Yeah.
some families have just sort of, we have equilibrium, and
Yep.
It's not, you don't want to force people to do something they're not interested in doing, but, include them.
Yeah, absolutely. And there are some families who have kids who are stable and healthy and relatively seizure free and not having terrible sleep problems or agitation or behavior, and they're fine. And so I get that they don't want to take a lot of risks. That's understandable. If you have come to terms with everything and your kid is overall healthy and happy, then why rock the boat?
Yeah.
But that's definitely not everyone.
Yeah. Well, any last comments from anybody? Floor is open.
It just seems to me that you've amassed a really cool team of people here who are all coming at the problem from different directions, and sounds like a whole lot's going to happen in a short period of time in the future, and congrats.
I sure hope so. Thank you. I just appreciate all the people that I mean, with the holidays and everything, those registration invites went out pretty late. And like you, Dr. Weston, I appreciate you jumping on board at the very last minute. So thank you all so much for your time. And I'm really excited about the connections that have been made today and where we're going to take this going forward.
Well, great job, Michelle. You pulled it off. You can go have some rest the rest of the day.
Yeah, for sure. And you, like I said, you were very, very central to it happening. So
I just pushed. Listen,
Yeah.
yes, thank you everyone for participating too for this.
Have a great rest of your weekend, everyone.
Okay.
Bye-bye.
Bye.
Hi, everyone.
Thank you. Bye.